Welcome to Genetics 2, Types of Gene Action. The narrator is Dr. Tony Sakura. And as far as types of gene action, we're going to be going over additive. And there's three types of dominance: partial, complete, overdominance. And then the other type of gene action we'll be going over is epistasis. With additive, each gene, or allele, if you will, acts independently. So in our examples here, we're going to use A loci. We're going to have big A and little a. And in this little equation here, we're saying that the value of the heterozygous genotype is the average of the two homozygous genotypes. So just as illustration, we're going to assume the big A is a 3, the little a is a 1. Now, where did these numbers come from? We just made these numbers up as an illustration. So with A equal to 3, it could be additional 3 pounds of milk a day. It could be that a rat with a big A allele is going to grow 3 ounces more over a 30-day period. This could be equal to a trainability score on a 1 to 25 scale. So these numbers are just made up as an illustration. They came from nowhere. It's pretty easy to determine the value of a genotype because we just look at the two individual alleles here. So each A is equal to a 3. So we got 2 big A's here. 3 plus 3 is 6. The homozygous dominant genotype has a value of 6. And again, that could be additional pounds of milk. It could be additional grams of daily gain. Now the heterozygous individual, we got a big A at a 3, a little a as a 1. Just going back here, getting those values. So that equals 4. In the 2 little a's, we got 2 little a's. They're each worth 1. That gives us a 2. An important thing to realize here is that the heterozygous is the average or midpoint between the two homozygous genotypes. And this is illustrated by this graph here. So on the x-axis, we have our 3 genotypes. On the y-axis, we have our values. And notice that we could draw a straight line here, and the heterozygous individual midpoint between the two homozygous recessives, or two homozygous dominant and recessive. With dominance, the pairs of alleles at that loci is going to be determine the value. And the value of your heterozygous individual relative to the homozygous genotypes determines the type of dominance. Whether that dominance is going to be partial or incomplete, whether it's going to be complete dominance or overdominance. So with partial dominance, and sometimes known as incomplete dominance, the heterozygous individual is less than the homozygous dominant, but it's going to be more than the average of the two homozygous genotypes. And that is illustrated here. Heterozygous individual closer to the homozygous dominant than it is the homozygous recessive. So if we have a value in this range here, anywhere less than here, but more than the average of the two homozygous genotypes, anything in this area here, if we have a value in here, we say we have partial dominance or incomplete dominance. Complete dominance is probably the easiest to illustrate and to understand, and basically your heterozygous individual is going to be equal to the homozygous dominant. You cannot distinguish between these, either numerical or by looking at the animal, whether if the animal has a homozygous dominant genotype, it could also be heterozygous. You cannot tell these apart. And they're going to be different than the homozygous recessive. So illustrated here, we've got two values, homozygous dominant, heterozygous, same value here. And that is illustrated by our graph here again. So we could draw a line right straight here. We're at this point here. This illustrates complete dominant gene action. So here's an illustration often used for in animal production classes to illustrate complete dominance. So the black hair pigmentation is generally dominant over red. So if you got one copy of the black pigmentation, it's going to mask any expression of this red allele here. Another example that we use, and it kind of depends upon species, but in a Bos Taurus cattle, polled is dominant over horned. So we have the horn gene here, and the horn is generally the wild type, and the wild animals are going to be horned, and they evolved that way. But a mutation came about thousands of years ago, and with that mutation, the animals are polled. And if you have that mutation, whether you have the horned allele or not, the animals are polled. You only need one copy of that polled allele for the animals to be polled. So overdominance, with overdominance, the heterozygous individual has an advantage. And again, we just kind of made up these numbers here as an illustration, but put an equation form here. The heterozygous individual greater than the homozygous dominant, and you know, these could be equal. The homozygous individuals could be equal, or homozygous dominant could be have a higher value. But basically what we're saying here with overdominance is your heterozygous individual is best. So this is just illustrated by the graph again. So here we have the value of the two little a's. Here's the value of the two big a's. So for overdominance, we have to be in this area here to say that we have overdominance gene action. It has to be better than either of the two homozygous genotypes. As far as examples of overdominance in animals, there are very, very few examples, and there may not be a lot of cases where we do have overdominant gene action. But here's a pretty clear example in Inverdale, the Inverdale gene in Romney Sheep. So we have 3 genotypes here: homozygous dominant, heterozygous, homozygous recessive. We're looking at ovulation rate. Ovulation rate is how many eggs on average are ovulated from the ovary during the breeding season. If we have 2 big eyes, average of 1.7. And your average lambing rate then is going to be somewhere between 1 and 2, maybe 1.5 on average. If we have a big I and little a, we're going to have 1 additional egg ovulated. And that's definitely an advantage if you want a lot of twins born. So you're going to get almost 1 additional lamb born if they're heterozygous. Now, if the homozygous recessive, that's a really bad condition, so they're going to be infertile. You're not going to get any lambs. So these little 2 eyes are kind of self-defeating in your ewe flock. They're going to disappear pretty fast. So you definitely don't want ewes with 2 little eyes, but big eye, little eye, heterozygous individuals the best. That's what we call overdominance. Now here's an example of overdominance in humans, and it's a little complicated because it involves 2 different traits. So trait number 1 is if you're going to be susceptible or genetically resistant to malaria. And of course, malaria is a parasite disease that you find in the tropics. And it's a bad disease, and thousands of people worldwide die from malaria every year. Well, this gene also affects the formation of your red blood cells. So sickle cell anemia just means that the red blood cells are not formed properly and have a harder time and are not very efficient as far as transporting oxygen to your organs that need it. So if you're 2 big A's, you got normal blood, but you're susceptible to malaria. If you've got 2 little A's, you're resistant to malaria, but you have single cell anemia, which is not necessarily a good thing. So the individuals that are heterozygous here have the clear advantage. They're going to be resistant to malaria, and they're going to have normal red blood cells. Just another little quick illustration in humans. The Rh blood group, you have probably heard that before as far as how it might affect the baby. If the baby or fetus is Rh positive and the mother is Rh negative, in humans, the mother's immune system might actually start attacking the blood group of the fetus. So it's important to get tested for that. But we also have a case here of interaction between this Rh factor and the D loci. So here we have, if we have this genotype, we're Rh positive, we got short-term protection against this parasite that causes toxoplasmosis. If you're Rh negative with this genotype, we have no protection. If you're Rh positive with a heterozygous individual, well, we have long-term protection. You're basically pretty much immune to toxoplasmosis. So again, the heterozygous in this case has the clear advantage. Epistasis is another type of gene action. Previously here we were talking about interaction of genes and value of the genes at a loci. Epistasis, we have interaction between genes at different loci. And probably there are definitely a lot of examples where different genes have to work together. So we got the example here. We got coat color in cattle, same for other species as well, I think. If you got one big E, we have black hair. Two little E's, you're going to have red hair. And then in a lot of species, including humans and rabbits and squirrels, we have a case where at the C loci, two little C's will totally restrict any pigmentation at all. The hair is going to be white, eyes are going to appear pink. So two loci working together here. Whether you have black hair or red hair pigmentation, genetics for that, if you get the two little C's, the E loci is not going to be expressed. So the C loci can totally interfere with what's happening at the E loci, and we call that epistasis, where one loci, genes at one loci will interfere with or change the expression of genes at another loci. So here we have another just simple little example, I guess. We said the two big E's or two big E's, or if you just have one copy of the big E, you're going to have black pigmentation. However, there's another loci, the A loci, that if you got a big A, just need one copy, it's going to restrict black hair to the extremities. So here we have the Bay as the genotype for black hair, but because we've got a big A here, the only place you find the black hair is at the extremities. So we got a black mane and black ears. We got a black tail. We're getting black down here. There's also another loci that will code for these white stockings. Well, if you got the gene for the white stockings, you're going to have white on the lower feet, regardless of what you have elsewhere. It may also account for the white blaze that we have down the face here. So we're actually illustrating the expression of genes at three different loci in this particular example. So here's a little practice problem for you on gene action, and in this for this particular class, we'll usually quiz the students, or you might find an example very similar to this on the exam. So take a minute to look at it, and your choices here. It's either going to be additive, partial dominance, complete dominance, overdominance, and that's our choices. You have four choices here. Epistasis is not illustrated here because with epistasis you need genes at two different loci. So just additive, partial dominance, complete dominance, overdominance. You might notice that the homozygous genotypes are the same in all cases here. And what's changing is the heterozygous values here. And it's the relationship between the heterozygous genotype values with the homozygous genotype values that determines the type of gene action. So again, we got four different types here: additive, partial dominance, complete dominance, and overdominance. Which is which? And here are our examples. Here's the answers. So complete dominance, these values are the same. With partial dominance, the heterozygous is going to be close in value usually to the homozygous dominant, more than the average of the two homozygous individuals. Additive, the heterozygous is the average of the other two. So we got a 1 plus a 5 is a 6. Divided by 2 will give us the average, which is 3. In overdominance, the heterozygous individual is the best or has the highest value. So that completes the little video on types of gene action. Have a good day.